In this session, we will discuss about the T-Lymphocytes production, as well as structure of thymus, which is primary lymphoid organ responsible for development and maturation of T-lymphocytes. As we have already discussed that lymphopoiesis is the production of new lymphocytes from the bone marrow. This is the bone. Inside the bone, there is a bone marrow, and inside the bone marrow, there are stem cells. These are called as pluripotent stem cells. In the production of lymphocytes, some of the pluripotential stem cells become early lymphocyte progenitor, and then lymphopoietic stem cells, which are also called as common lymphoid progenitor, that will move to thymus and forms thymocyte and then complete its development as mature T cell. The mature T cell, having gamma and delta, will be expressed as gamma and delta T cell receptor. But T cell having alpha and beta will be expressed either CD4 or CD8, then they are released in the bloodstream. Although the fundamental requirements for differentiation of B and T lymphocytes are similar, the situation with T cells is more complex than with B cells for two reasons. All B cells produced in the bone marrow are the same except for the specificity of their surface immunoglobulin M and immunoglobulin D. By contrast, three types of T cells are produced in the thymus from the same type of precursor cell. Two of these cell types are the CD4 and CD8 T cells, expressed as alpha beta T cell receptor. And the third type bears a different receptor for antigen, the gamma delta T cell receptor. All of these cell types are produced in the thymus. The second reason why T cell development is more complicated is that T cells recognize antigen in association with MHC. T cells must be selected in each individual to recognize antigen in association with that individual's own MHC, a process known as Thymocyte education. To understand the concept of alpha, beta, gamma, and delta T cell receptor, watch our video on T cell receptor. Now, let's understand the structure of thymus, which is the primary lymphoid organ. The thymus is located in the mediastinum and lies above the heart. It has a two-lobed structure, and each lobe is divided into many lobules separated by trabeculae of connective tissue. Each lobule has two sections that can be easily distinguished histologically. The thymus reaches its peak size around 1 year of age, after which it shrinks or involutes. In adults, it is composed mostly of fat cells and connective tissue, although it produces new T cells well into old age. Numerous different cell types can be found in the thymus, which all play a role in T cell development. Let's discuss their role and structure. The epithelial cells. Three types of epithelial cell have been described in the thymus. First type is the cortical epithelial cells. These cortical epithelial cells are the type of cell that make up a mesh throughout the cortex, so that cortical epithelial cells provide structure. And another important function of these cortical epithelial cells is that they also secrete factors that are essential for T cell development. Second is nurse cells. These nurse cells are second type of cortical epithelial cell that can be seen in close contact surrounding developing thymocytes. These are the type of cells that express MHC class I and MHC class II on their cell surface. Third is medullary epithelial cells. These medullary epithelial cells provide structure for the medulla, and recent evidence suggests they play an important part in T cell tolerance. These are the macrophages. These cells, which are macrophages, are found in both the cortex and the medulla. Although they are more numerous in the medulla, they are class II MHC negative, and macrophages play an important role in phagocytosing thymocytes that have died by apoptosis during development. Now, These are dendritic cells. These are another type of bone marrow-derived cell related to dendritic cells found in most tissues and lymphoid tissue. They express class II MHC as well as class I MHC. These are the Thymocytes. These are the most abundant cells in the thymus and consist of T cells in various stages of development. This is the Hassel's corpuscle, a structure peculiar to the thymus, in which epithelial cells become concentrically compressed and keratinized, possibly the site of removal of apoptotic cells. Although the function of Hassel's corpuscle remains unclear, it may have a role in the production of Treg cells, which are regulatory cells that help to maintain tolerance to self. The outer area stains more densely with hematoxylin and eosin and is called the cortex. The less densely staining inner section is called the medulla. It should be mentioned that there is no apparent anatomical barrier between the cortex and medulla, although particulate material injected intravenously is able to access the medulla, but not the cortex. Now moving towards the selection of T cells. Because of its importance and complexity, the process of selecting T-Lymphocytes for export has attracted intense study and is currently considered to consist of the 5 stages. Stage 1 is that CD4 negative and CD8 negative cells proliferate in the outer region of the cortex, during which they become CD4 positive and CD8 positive, and rearrange their TCR genes. Stage 2 is that under the influence of thymic stromal cells, T-lymphocytes, whose TCR recognizes one of the available self-MHC molecules, survive and the rest die. Stage 3 is that cells that recognize an MHC class I molecule lose CD4 and retain CD8, and those that recognize an MHC class II molecule lose CD8 and retain CD4. Stage 4 is that under the influence of dendritic cells, presenting self-antigens in the form of short peptides, potentially self-reactive T cells are eliminated. Then stage 5 is that the remainder, probably only about 2% of the starting population, are allowed to exit, and these make up the peripheral T lymphocyte pool. This was all about the production of T cell, as well as detailed explanation of structure of thymus. In our next session, we will talk about the B cell production and detailed explanation of structure of bone marrow and bursa of Fabricius.